Biology 1107, Presentation 5, Membranes. This presentation concerns biological membranes. Biological membranes are membranes found all throughout the cell. They can surround the cell, they can surround the individual nucleus, they can surround individual organelles, they can be found inside organelles, they can surround tiny little sphere-like structures that are used for transporting substances in the cell. Those spherical structures are called vesicles. Membranes can be found all throughout the interior of the cell, interspersed in a network throughout the cytoplasm. So biological membranes are found virtually everywhere. I even find them in bacterial cells. So what do? Well, essentially a membrane is going to be an interface. It's a boundary between one place and another. So, for example, the cell membrane is a boundary or an interface between the cytoplasm and the environment around the cell. The nuclear membrane is an interface between the nucleoplasm, the content of the nucleus, and the cytoplasm around the nucleus. Even within the mitochondrion, there are membranes that separate the mitochondrion into compartments, and those compartments necessarily need a boundary between them. Now, the thing is, in many cases, that boundary is going to have to provide us some exchange function. In the case of a cell membrane, there are food items I might want to bring in, maybe carbohydrates, maybe amino acids, maybe ions, and there's some things I want to get rid of, like waste molecules. So exchange is going to be an important aspect of the function of a biological membrane, at least in terms of it being an interface. So what are some of the other functions? Well, the other functions include cell recognition. That particular function is largely limited to the cell membrane itself. It's how your body actually is able to distinguish your cells from cells that might not belong. You all have probably heard of transplant rejection. So when somebody donates another individual a kidney, if that kidney does not match the recipient's body is going to fight it off. It's going to regard it as being foreign. And that boils down to the recognition of some certain proteins found on the cell membrane. Bottom line is the cell membrane is where the rubber hits the road in terms of being able to recognize a cell as being self or non-self. Kind of as part of our body defense system. Membranes can be places where we anchor enzymes, not only in the surface of the cell, but all throughout the cell. You remember enzymes are proteins that have certain jobs to do. And it might be nice to have an enzyme anchored in a particular place. I maybe might want to anchor ribosomes. Ribosomes are this tiny intracellular structures that crank out protein. And then finally, probably the most important thing that biological membrane can do is establish a gradient or a concentration difference. If a membrane is going to be an interface, what I could conceivably do is I could create a high concentration of some particular molecule on one side of the membrane and a low concentration of some molecule on the opposite side of the membrane. And as a consequence, use that gradient or that concentration difference as a mechanism to motivate the movement of molecules across that membrane, generally from a higher concentration to a lower concentration. But having said that, we know that biological membranes are found all throughout the cell. They function as an interface. They have a number of other functions that we will talk about in greater detail on the next series of slides. No one has ever seen the structure of a membrane. As strong as some electron microscopes are, we don't have any pictures that effectively show us the arrangement of molecules in a membrane structure. However, based on our knowledge on how those molecules might behave, and based on our knowledge of the composition of membranes, we can come up with a pretty good idea that explains how a membrane is put together and also explains how a membrane might behave or function in some way, shape, or form. Now, this idea of what a membrane actually looks like is called the fluid mosaic model of membrane structure. It's a basic idea that a membrane is a composition of a lot of different molecules. Several different types of molecules, in fact, and that's where we get the word mosaic. And fluid is the idea that the molecules are not physically connected to one another. There's some degree of attraction. However, they do demonstrate a certain degree of fluidity. That is to say, they move, and they are not physically fixed to one another. And a lot of this is based on what we know about those molecules that make up membrane structure. So I've got a real rudimentary illustration of a view of a membrane in cross-section here, and you'll see some basic components. There are two basic components found in a membrane's structure, and it doesn't matter whether it's a cell membrane or a membrane in a mitochondrion or a membrane surrounding a bacterium. They all have the same basic structure, and that is phospholipids and proteins. And the phospholipids are going to be consistently arranged in a bilayer. Now, you'll remember phospholipids behave this way because they are amphipathic molecules. Phospholipids have a split personality. They love water, at least the head portions, and they hate water, at least the tail portions. And in an aqueous or water-based solution, phospholipids will always form a bilayer as illustrated here. The blue circles being the head portions of the molecule, which are hydrophilic, and the straight lines being the tail portions of the molecule, which are hydrophobic. Now, interspersed in this bilayered arrangement are proteins. Now, what I'm just going to say is while phospholipids, and that is misspelled on the slide, by the way, but phospholipids are the primary structure of the membrane, but most of the functions of the membrane lie with those proteins. Proteins are critical to the functions of the membrane. So let's make a comment about fluidity here. Now notice something. Notice the phospholipids are rather small and the proteins are rather large. It's kind of a general rule of thumb. Even though phospholipids, if you look at them as a chemical structure, they appear to be large, they are nowhere near the size of these massive proteins. So if my understanding is correct, that these molecules are not physically fixed or connected to one another, they're going to move. And they are going to move at a certain rate. Now, the smaller molecules tend to move much more quickly. So you'll notice my bullet point says lateral movement of phospholipids is rapid. Those little blue balls with the double tails are going to be moved around. They will change places and will do so rather rapidly. Now, on the other hand, the proteins are capable of doing the same thing. They will move around, but they don't move around anywhere near as fast because they're much larger. So the lateral movement of proteins is slow. But now you understand, in your mind's eye picture, you can imagine a membrane as not being a structure of fixed molecules, but it's a structure of molecules that are constantly moving around. And temperature plays a huge influence on that. The higher the temperature, the greater the fluidity. The lower the temperature, the greater the viscosity. Now that word viscosity literally means thickness. So if you were to basically, dish yourself up some pancake syrup on a very hot summer day, the pancake syrup is very, very liquidy and runs very easily out of the bottle. But yet, if you do that on a very, very cold January morning, that same pancake syrup is going to be really, really thick and slow. Now, being thick and slow, that's what that word viscous would apply to. And the word viscosity, therefore, implies the consistency of a liquid, whether it's going to flow quickly or flow slowly. So as temperature goes up, viscosity goes down. As temperature goes down, viscosity goes up. Now that does present a problem for the cell if it's going to depend on its membrane to be functional. In other words, if I'm going to depend on my membrane to let things in consistently, to let things out consistently, and at the same time I want to depend on my membrane as having enough integrity to maintain the outside of my cell or the outside of my organelle, I need to maintain some consistency of viscosity, which means I need to have some way of being able to resist the effects of temperature as it goes up, which increases the instability of the membrane, or if it goes down, which decreases the fluidity of the membrane. Both of those could be problems. So there is a third component I am going to find amongst the phospholipids and the proteins in my membrane structure, and that's cholesterol. Cholesterol itself is not a very large molecule, but cholesterol has a moderating influence on temperature. So therefore, if temperature goes up, and I would expect some increase in fluidity of my membrane, and I would expect some degree of instability of my membrane because the temperature is going up and viscosity is going down, cholesterol resists that. The cholesterol resists the decrease in viscosity. The cholesterol resists the increase in fluidity. The cholesterol maintains the stability of the membrane in the face of temperature rise. Now, on the other hand, if temperature falls, I certainly don't want my membrane becoming so terribly thick and viscous that it's going to be unmanageable. Well, cholesterol deals with that too. So as temperature goes down, cholesterol makes sure that there's a certain degree of fluidity in the membrane as well. So understand that cholesterol is going to be key to being a mechanism of, I guess we could use the word homeostasis with respect to membrane fluidity. Just because I can't resist it, I've got to post a slide here about phospholipids. This is nothing more than a review from presentation number 3 on organic chemistry. Just make sure you remember that a phospholipid is a lipid, and it has a structure that is very, very similar to triglycerides. Big difference is instead of having 3 fatty acids, there are 2 fatty acids bonded to the glycerol molecule, and in place of the 3rd fatty acid, there's a phosphate group. The phosphate group has a negative charge. As a consequence, that portion of the molecule called the head is polar, is hydrophilic, that is water-loving. The tail portion of the molecule, the fatty acids, are nonpolar, are hydrophobic. And in aqueous solution, in water, the phospholipids will form a bilayered arrangement, which constitutes the primary structure of the membrane, as we saw earlier. The phospholipid bilayer is critical in determining whether molecules can pass through the membrane structure or not. As a consequence, nonpolar molecules, that are molecules that don't have any charge, are capable of passing through the membrane. So nonpolar molecules would include hydrocarbons, would include carbon dioxide, would include oxygen, and these molecules can pass through the phospholipid bilayer. They just simply slip in between the phospholipid molecules. Now, when a molecule can pass through a membrane, the membrane is said to be permeable to that molecule. Now, you'll notice that the title of my slide indicates here that there is a selective permeability demonstrated by our membranes, and that means they're kind of selective in terms of what can pass through. Sometimes the membrane will be permeable to a particular molecule, like our nonpolar molecules, like our hydrocarbons or carbon dioxide and our oxygen, but sometimes the membrane might be impermeable to the passage of molecules, and those would be the polar molecules that I see in the second bullet on our slide. Polar molecules are charged molecules, and if you've got a charge on you, positives or negatives, or if you're a larger molecule, you will not pass through that phospholipid bilayer. So polar molecules include water, include sugars, include ions, and these are molecules that will not pass through the phospholipid bilayer. The bilayer, or the membrane, would be said to be impermeable. So make sure you are clear on the following terms. The word permeable means the membrane allows a molecule to pass. The term impermeable means the molecule will not pass. And selective permeable or selective permeability means that some molecules can and some molecules cannot. Now, I will mention there is a synonym to the idea of selective permeability, and that is the membrane can be said to be semi-permeable. But make sure, as far as content goes, you know what sorts of molecules can pass through the phospholipid bilayer and what molecules cannot. Well, just because a molecule is polar, or just because a molecule is big, doesn't mean that that molecule does not have an avenue of entering or leaving the cell through membrane. So what we're going to find is there are some additional mechanisms by which we can actually get around the idea of the membrane being impermeable. Case in point, water. You will see in our illustration on the upper right, you will see a section of membrane. You'll see a big fat protein illustrated there in pink, and you will see an opening in the middle of that protein allowing the transport of water from one side of the membrane to another. Water is a polar molecule. Ordinarily, water would not be able to pass through those phospholipid molecules, but it does. The passage of water molecules across the membrane is absolutely critical to the survival of the cell. So what we're going to do is we're going to find out there are special proteins called channel proteins that are going to function in the passage of water. You can think of them as like gates. A channel protein is a passive gate. It will allow water to pass through the opening in it, and that basically is a case for what we will be talking about in a bit called osmosis. Osmosis is the movement of water across a membrane, and that's what we're looking at, this illustration on the upper right. Now, these specialized channel proteins that specifically allow the passage of water are called aquaporins. Aquaporins are channel proteins that are specific to the movement of water across the membrane. Now, on the other hand, some proteins change their shape when they afford movement of a molecule, and those are called carrier proteins. Carrier proteins are proteins that will change their shape but work in much the same way. You can think of a channel protein as a permanently open gate, whereas a carrier protein might open and shut, and the opening and shutting of that carrier protein or the function of the carrier protein sometimes occurs with or without the input of energy. So I am looking at an illustration here, the bottom right, and you'll see this is called the sodium-potassium pump. Details are not important. This is something, though, that you will encounter in Biology 1108 at some point. But what I see is the artist's rendition shows me the phospholipid bilayer. I see a protein on the left. I see the same protein on the right. And the protein on the left shows me that I am going to be moving sodiums from the inside of the cell, and I am bringing in potassiums from the outside of the cell. And if I shift my attention to the right, at that point, the sodiums are on their way out and the potassiums on the way in. And then you will see that those two proteins are accompanied by the input of some ATP, which is energy. So energy is essentially responsible for our function of that carrier protein doing its job. Now, a lot of times, carrier proteins are sometimes also referred to as pump proteins, specifically if ATP is invested in a process. But please understand that sometimes a carrier protein will just simply change shape just by virtue of the fact that there are some molecules that need to come into or out of the cell. I'm thinking, for example, of a revolving door. If you think about revolving doors, and when I was a kid, we used to play with them. If you have enough people pushing against a revolving door, the revolving door will turn and let people in or let people out without any further addition of energy. But on the other hand, if you want to go into or out of store and the automatic door senses that you are there, energy is used to cause that door to slide open, and you can enter the grocery store or enter the bank or whatever building you want to get into. So essentially, make sure you're aware of the fact that proteins do play a role in transport. There are channel proteins, which are permanently open gates. There are carrier proteins that can change their shape. If they change their shape in the presence of energy, they are sometimes specifically referred to as pump proteins. I mentioned not too long ago that proteins are responsible for many of the functions of a membrane. So, got to understand that's not just limited to the function of transport. So I do have transport proteins mentioned at the top of this slide, but proteins do a lot more than just transport. There are enzymes present in an enzyme structure, and those enzymes are proteins that are specifically devoted to accomplishing certain biochemical tasks. There are signal receptors and transducers, and these are going to be proteins that actually receive information from external molecules like hormones, and that information is then conducted into the cell, and that's transduction. And so there are proteins in the membrane that will actually transmit that message into the cell, much like a person whispering a message to another person, whispering a message to another person, getting that message into, for example, a room to a specific individual. Then there's cell recognition, that cells do have a way of essentially displaying their identity. Primarily, it's self versus non-self. I mentioned that not too long ago. And so in the surface of the cell membrane, I've got specific molecules that engage in that process. Those specific molecules include glycoproteins. Now, glycoproteins are proteins that have a carbohydrate chain attached to them. And so you can think of a glycoprotein as being embedded in the membrane structure, and the carbohydrate chain is displayed to the outside of the cell. So in many ways, this carbohydrate chain is a chemical name tag that is identifying the cell as to who it belongs to. So for example, your cells, my cells, have our own unique assemblage of carbohydrate chains being displayed to the outside of the cell. And those chains are ultimately attached to proteins. Those molecules specifically are called glycoproteins. Now, there are sometimes lipids that look almost the same. A lipid can be embedded in the cell membrane, and it can have a carbohydrate chain as well. And so lipids featuring carbohydrate chains are called glycolipids. And glycolipids give us the same basic function. A glycolipid will have a carbohydrate chain which will function as a chemical name tag. So the complete assemblage of carbohydrate chains that identify your cells as belonging to you or my cells as belonging to me are a combination of glycoproteins and glycolipids, both embedded in the cell membrane structure. I see intracellular linkages here. What causes cells to be connected to one another? Well, we talked about this in the context of the presentation on cells, and we talked about a variety of intercellular connections, like gap junctions and tight junctions and desmosomes, and a lot of times those intercellular connections are mediated by proteins. Cytoskeletal linkages means that the cytoskeleton can be ultimately connected to the membrane. Think of the membrane as being stretched out over the surface of the cytoskeleton that gives the cell its unique shape. And at some point, or at least at multiple points, all throughout the membrane, there are going to be places where the membrane is literally snapped down onto or connected to the cytoskeleton below it. And then finally, outside of the cell, there's a whole huge hodgepodge of stuff. A hodgepodge of protein molecules, a hodgepodge of fat molecules, a hodgepodge of carbohydrate molecules, and a lot of times those external molecules are actually fastened to proteins in the cell membrane. All right, on the next few slides, we're going to be taking a look at passive transmembrane transport, How do we move molecules across a membrane structure without an investment of energy? That's the concept of passive transmembrane transport. The word passive implies no expenditure of energy. So what is going to be our mechanism? What's going to drive the movement of these molecules? What will drive the movement of the molecules is something called gradient, also known as concentration gradient. If you see our illustration on the top right, you will see a large number of molecules on one side of a membrane. You will see a series of arrows indicating the movement of those molecules across the membrane. Now notice the molecules are moving from the high concentration to the low concentration. And that all depends on what's commonly referred to as the kinetic movement of those molecules. Now, molecules in the natural world will always move, and they demonstrate what's called random kinetic movement. Now, that random kinetic movement is a lot greater if the molecules exist as a gas, maybe not quite so much if the molecules exist as a liquid, and not much at all if the molecules represent a solid. But if we're talking about molecules crossing a membrane, those molecules are primarily going to be in a form of molecules dissolved in water. So here I've got molecules dissolved in water, and they are going to be moving. They're demonstrating random kinetic movement. As temperature goes up, random kinetic movement increases, but it always happens, which means in your mind's eye picture, you imagine those molecules as moving and bumping into each other, and as they move and they bump into each other, sometimes they will actually cross the membrane. Now, understand something. In order to cross that membrane, the membrane has to be permeable to those molecules. So by virtue of colliding into one another, because of the crowded nature on one side of the membrane, those molecules will scoot their way in between those phospholipids. And so the goal of this transmembrane transport is what we will call a dynamic equilibrium. Compare the illustration at top right with the illustration at bottom right, and you will see that we now have an equal distribution of molecules and equal concentration above the membrane and below the membrane, and yet the arrows still indicate some degree of movement. That's why we call this a dynamic equilibrium. The word equilibrium implies, of course, that we have an equal concentration, but the word dynamic indicates to me that movement is still possible. So we could say that in this case, once I have achieved dynamic equilibrium, there is no net movement. I can't say there is no movement at all, and I certainly can't say that all the molecules have moved from one side of the membrane to the other, because that's not the case. But in this case, I have no net movement, which means if I am going to maintain this equal concentration, every time one random molecule moves from one side of the membrane to the other, some other molecule is going to move in the exact opposite direction. So in the next couple of slides, what we're going to be doing is taking a look at two specific types of passive transmembrane transport. And because we've covered this information about concentration gradient and the notion that this is a passive process and the notion of the random kinetic movement of molecules, the next two slides should go by rather quickly. We are going to document something called simple diffusion, and then we're going to document something called facilitated diffusion. Our first example of passive transmembrane transport is simple diffusion. Definition: simple diffusion is the movement of solute molecules directly through the phospholipid bilayer. Now, a solute is something that dissolves in a solvent. So the little yellow balls on our illustration here are solute molecules, and we'll say that those solute molecules are capable of passing through the membrane. So they might be small. More importantly, they're nonpolar. We mentioned this not too long ago. If you are small, if you're nonpolar, and you pass through the phospholipid bilayer, you are technically a penetrating solute. That's what bullet number 2 implies. Simple diffusion requires penetrating solutes, molecules that can pass through a phospholipid bilayer. No transport proteins are required in this case. It is a passive process because it is essentially driven by concentration gradient. As a matter of fact, you might want to scribble that down next to passive process. Concentration gradient, the molecules are moving, random kinetic movement, they're colliding, bumping into one another, and once in a while they will move from the highly concentrated region across the membrane to the less concentrated region. Follow the arrows. And once we're done, we have a dynamic equilibrium that results. So opposite, no energy is being expended in this process. And the conclusion of that is a dynamic equilibrium in the sense that we do have an equal concentration on either side of the membrane. However, it's a dynamic equilibrium in the sense it's an equilibrium in constant potential adjustment. Anytime one molecule moves north, another molecule moves south. Hence, the double-headed arrows. And we mentioned the fact that technically this is a situation where we have no net movement. The implication is, of course, that movement is still possible, but overall there is no net movement to one side of the membrane or the other. Make sure you know what are the hallmark features of simple diffusion. You're going to need to contrast them with the hallmark features of facilitated diffusion, which we'll see on our next slide. In contrast to simple diffusion, we have facilitated diffusion. In this case, we've got molecules that are moving across the membrane driven by concentration gradient. However, those molecules cannot pass through the phospholipid bilayer, so we need some help. That's where the word facilitated comes from. The word facilitate means to help. So let's take a look at this. Our molecules are non-penetrating solutes. Now, in this case, these molecules are polar or larger. They cannot pass through the phospholipid bilayer, but the green arrows indicate to me they can. So what's the difference? Well, the difference is I've got channel proteins or carrier proteins involved. Now, we mentioned them not too long ago. These are proteins that function like gates. Those gates can be permanently open in the case of channel proteins, or can open and close in the case of carrier proteins. And in this case, if the carrier protein requires no input of energy, then technically it's a passive process. So facilitated diffusion, non-polar, non-penetrating solutes, channel proteins and carrier proteins involved. It's a passive process, no energy involved, which means concentration gradient is our motivating mechanism. Large concentration on one side of the membrane results in a dynamic equilibrium, that is an equal distribution of molecules on either side of the membrane. However, because those proteins still exist, those channel proteins and those carrier proteins, the potential is that for every one molecule moving across the membrane in one direction, there will be one molecule moving in the opposite direction. Hence, we will go ahead and say this is a case of dynamic equilibrium, equal concentration, and no net movement. Be sure you're able to contrast simple diffusion with facilitated diffusion. So what happens if you want to move a solute molecule against a concentration gradient? There's a number of cases where this might be a necessity. Now, supposing I've got waste molecules building up inside the cell, and I need to move those waste molecules to the outside of the cell, yet, on the other hand, there might be a greater concentration of waste molecules on the outside of the cell, and therefore it would be very, very difficult for me to move a molecule against that concentration gradient. Well, a mechanism does exist, and it's called active transport. Active transport, the word active, would imply an investment of energy needed. So let's take a look at the hallmark features of what is active transport, making sure that we know that this is something that is completely, totally different from what we see in the passive transport forms that we saw earlier, simple diffusion and facilitated diffusion. So in this case, the big difference is we are moving molecules against a concentration gradient. Illustration top right. Large concentration of molecules, arrows indicating the movement of molecules not with the concentration gradient, but against. So we're moving from a low concentration to a high concentration. In my heart of hearts, I know that the random kinetic movement of those molecules would tend to encourage their movement from the top portion of the illustration to the bottom portion of the illustration. However, the arrows are indicating me in opposite direction. So this is going to require an investment of energy, and that energy is going to be in the form of our cellular fuel molecule, ATP. A carrier or pump protein is going to be required, hence the little red ovals there. It's going to be a protein that will literally grab hold of the solute molecule and force it, push it from the area of low concentration to the area of high concentration, and the result is going to be to maintain a concentration difference. That is a disequilibrium. A disequilibrium, technically, is nothing more than a term meaning the absence of an equilibrium. So we are going to reinforce the fact that we have a high concentration of molecules on one side of the membrane, and we're going to keep it that way because we use ATP as our fuel to activate these pump proteins to actually move the molecules from the lower concentration to the higher concentration. Now we're going to turn our attention to a unique form of transmembrane movement, and that is the movement of water molecules across the membrane. The official term for that process is osmosis. It's something we mentioned a little bit earlier in this presentation. Now, water will move across a membrane generally driven by tonicity. Now, the word tonicity comes from the word tonic, which means concentration. It is the concentration of non-penetrating solutes. Those would be dissolved molecules that cannot pass through the membrane. If you take a look at our illustration to the right on this slide, you'll see a lot of big purple balls. Those are non-penetrating solutes. Those are solute molecules. They're dissolved in water. The water molecules are illustrated as the small, little blue balls. And so they're dissolved in water, but the fact of the matter is the water molecules can pass through the membrane on account of those aquaporins we mentioned not too long ago, but those big purple molecules cannot. So if the purple molecules cannot move to achieve a dynamic equilibrium, the water molecules will. So if we take a look at the arrows, you'll see that I've got arrows going in both directions. That means that water molecules conceivably can move from the lower part of the diagram to the upper part of the diagram, or conversely can move from the upper part to a lower part. But the far majority of those arrows are illustrated as being larger than the others, so the larger arrows are indicating to me that I've got this net movement of water molecules from below the membrane to above the membrane. And what's driving that? It's the high concentration of the solute molecules that lies above the membrane. So that's my net direction of movement. Now, osmosis is the movement of water molecules across the membrane driven by tonicity. Now, there are three terms that I need you to know that are going to describe this relative degree of tonicity. The terms are hypertonic, hypotonic, and isotonic. Now, the word hypertonic means a higher concentration. So what I see above the membrane here, that is the upper portion of the illustration, that is a hypertonic situation. Below the membrane, I see a hypotonic situation. That means a less concentrated situation. So hypertonic, greater concentration, hypotonic, lesser concentration. And if my water molecules are going to be moving towards the hypertonic situation, I think you can imagine that as time goes by, the hypertonic portion of the illustration would become more dilute, whereas the hypotonic portion of the illustration here, we become more concentrated because the water molecules are leaving the lower area and moving to the upper area. So at some point, you ultimately will get some equal concentration of solute molecules. That's the trend. So the movement of water molecules is going to tend to decrease the concentration of the hypertonic region and increase the concentration of the hypotonic region to the extent that we're going to get some balance in concentration. Hence, my third bullet point, the term isotonic, meaning the same concentration. Now, the tendency to drive water molecules across a membrane is referred to as osmotic pressure or potential. So it is the concentration of the hypertonic region actually drives the osmosis. The hypertonic condition is what constitutes the osmotic pressure or the osmotic potential. But regardless, once those water molecules move, we will achieve a dynamic equilibrium, and that would be an equal concentration of water molecules above and water molecules below. Now understand that concentration needs to be thought of not only in terms of the number of water molecules, but also the number of water molecules in regards to the number of solute molecules. But ultimately, we're going to get this equal concentration. But we're still assuming the water molecules can move. There are still these aquaporins that will permit the movement of water molecules from one side of the membrane to the other. So technically, I've got an equal concentration, but it's an equal concentration in constant adjustment because of that potential for further movement of the water molecules. On the next slide, we're going to take a look at how osmosis has a practical application to both animal cells and plant cells. And we're going to find out that basically water balance is something that's very, very important to cells. So we want to make sure that we balance the amount of water inside the cell versus the amount of water outside the cell. And we can actually drive the movement of water into tissues and out of the tissues because of osmosis. And that can have beneficial consequences, but if it runs out of balance, it can have negative consequences as well. So we're going to take a look at practical osmosis on our next slide. Let's take a look at practical osmosis. We'll take a look at practical osmosis in animal cells and in plant cells. And you will see a series of illustrations above for animal cells and below for plant cells. If you wanted to, you could conceivably Google osmosis and come up with all kinds of similar illustrations that will reinforce this content as well as the content on our previous slide. But on this particular slide, as we take a look at the practical influence of osmosis on animal cells and plant cells, we will apply those terms that we learned on the previous slide. We will apply the terms hypotonic, hypertonic, and isotonic with respect not only to the cells, but to the environment surrounding the cell. So I'm going to try to make sure that's clear because those terms, hypertonic, hypotonic, and isotonic, are all relative. They are relative to the extent that we're comparing the tonicity of the interior of the cell to that of the environment around the cell. That's why we say it's relative. If the interior of the cell is more concentrated than the exterior, we can say the interior of the cell is hypertonic, whereas the exterior of the cell is hypotonic. If the interior of the cell is less concentrated, we say that the interior cell is hypotonic compared to the hypertonic situation in the environment. See, they're all relative. So let's take a look at osmosis in animal cells. And the series of 3 illustrations here shows me a red blood cell. Now, we got millions and millions of red blood cells found in our bloodstream. And the central illustration shows us what a red blood cell essentially looks like. It is kind of a disc-shaped cell, has a depression in the center, and I notice that in the illustration here, we've got an equal amount of water moving in and an equal amount of water moving out. That cell is isotonic. It's isotonic with respect to the environment around it, and that's the normal case in our bloodstream. So all of our red blood cells are going to maintain their volume because they have an equal amount of water flowing in and amount of water flowing out, and that's driven by solute concentration, but those are equal. So essentially, I have what I would call an isotonic situation. Now, let's take a look at what would happen if instead of having an isotonic situation, let's suppose the interior of the cell is hypertonic. Let's take a look at the far right-hand illustration. In the far right-hand illustration, you'll see that the environment is illustrated as being a deeper blue than the other two, indicating a hypertonic environment or a hypertonic solution. At the same time, if the environment is hypertonic, got to admit that the interior of the cell is going to be hypotonic. So I've got a relative difference here. And I notice that the illustration shows me that water will be leaving the cell. The net movement of water is going to be out of the cell, primarily because it's being driven by the tonicity or the concentration of dissolved solutes in the environment around the cell. As a consequence, as the water leaves the cell is going to shrivel. It's going to decrease in volume, and that process is commonly described as crenation. So if you would circle the word crenation and draw an arrow over to the illustration that's labeled there as shriveled. Essentially, the cell loses water in a hypertonic environment, and crenation is the result. Now, let's take a look at the other hand. Far left-hand side illustration, what happens is the environment is hypotonic. It's less concentrated, which would mean the interior of the cell is hypertonic, which means the net movement of water is going to be into the cell, driven by the hypertonicity inside of the cell. But understand the cell is of limited volume, right? So as the water moves in, the cell is going to get bigger and bigger and bigger, and at some point, as the illustration shows me, the cell is going to burst. That is called lysis. So I would go ahead and circle the word lysis and draw an arrow to that leftmost illustration that is labeled as lysed, which shows me that this cell, having experienced an input of water driven osmotically by the concentration inside the cell, is going to get bigger and bigger and bigger and finally burst. And I mentioned essentially osmosis can have its benefits, particularly if we're maintaining an isotonic situation. However, can be detrimental if we experience crenation, or more significantly, if we experience lysis. Let's take a look at the situation in plant cells. Now, in plant cells, big difference. We have a cell wall surrounding the cell, which means when the cell increases in size, should osmosis occur, it's going to run into that cell wall. So the cell's inside of a box. So let's take a look at osmosis in plant cells. We'll take a look at the central illustration first. Once again, this is the isotonic situation. In an isotonic situation, the concentration of the cytoplasm inside the plant cell is essentially the same as the environment around the plant cell, which means my net water movement is going to be zero. You'll see the yellow arrow indicating the movement of water into the cell, and the green arrow indicating the movement of water out of the cell are equal. That's what we call a flaccid situation. The cell is not experiencing a tremendous amount of fluid pressure buildup inside. As a matter of fact, the concentration of fluid inside the cell is exactly the same as the concentration of fluid outside the cell, so therefore there is no net difference in fluid pressure. On the other hand, take a look at the situation to the far right. What happens if we have a hypotonic situation around the cell? If I have a hypotonic situation, that means the interior of the cell is hypertonic, which means that the water flow is going to be from the hypotonic environment into the hypertonic cell. You will see that what happens is the cell gets a little blown up. It's a little bit of what happens when you put air into a bicycle tire. Most of us realize that a bicycle tire consists of an inner tube contained inside of a hard rubber tire. And if you pump air into an inner tube, all by itself, that inner tube will get bigger and bigger and bigger. It will blow up like a balloon. But the inner tube is contained inside of the tire. So as you blow up the inner tube only expands so much before it runs into the hard rubber of the tire, at which point you increase air pressure in the tire, and the tire becomes hard and rolls fairly easily, which is what you need to have happen with your bicycle. If you take a look at our illustration here, I see as water moves into the cell, it's almost like pumping it full of air. The cell gets bigger and bigger and bigger. Now that is what is called turgidity. That cell is said to be turgid, and that turgidity is a consequence of increased fluid pressure inside the cell. So somewhere off to that far right-hand illustration, I would write that down. Increased fluid pressure due to osmosis. This is one of the reasons why on a practical level, when you buy yourself some fresh lettuce or you take a bite of a fresh stick of celery, you know, that piece of plant material is crispy. And when you bite into it or you break it, it's going to crunch. It's going to crunch and it's going to spray water about, which is one of those delightful things about vegetables. But the fact of the matter is that crispiness in the lettuce or that crispiness in the celery is a consequence of what we would call turgidity of those cells. We could say that those cells have a certain amount of turgor pressure. Turgor. So you could write down those words as well to the right-hand illustration. Turgor pressure. That's the fluid pressure that exists inside that cell. Turgor pressure is what makes for a turgid plant cell. Okay, so Dr. Knaus, what happens if the tables are reversed? Let's take a look at our left-hand illustration. If our environment is hypertonic, that means the interior of my cell is hypotonic. And that means the net movement of water is going to be from the inside of the cell to the environment. Illustration shows me green arrows indicating the movement of water out of the cell. But the cell exists inside of the cell wall, so what's going to happen is the cell is going to shrink inside. When a cell shrinks inside of a cell wall, that is commonly called plasmolysis. And now once again, there's benefits to osmosis in this case. The healthy plant cell is going to be somewhere between isotonic and hypotonic. By and large, the far majority of plant cells would much rather be the illustration over there on the right. It's going to be a cell that is turgid, that is demonstrating turgor pressure. On the other hand, if you lose water, as what I see is happening on the left, we call that plasmolysis, but that's nothing more than a practical result also of what you and I would call wilting. When a plant doesn't have enough water, what happens? Well, it wilts. It falls over, it shrivels up, and that's on account of not having enough turgor pressure in those individual cells. Be sure you know the difference between practical results of osmosis in animal cells and osmosis in plant cells. Know all those terms. And if it doesn't make sense here, Google for other sets of images that might be agreeable to you. Okay, for the far majority of our presentation here, we've been talking about transmembrane movement, moving things into and out of the cell through the membrane. What we're going to do now is we're going to take a look at how we can move large things into and out of the cell without necessarily passing through the membrane structure. That might not sound like it makes sense, but let's take a look at some illustrations here. The very first thing I want to talk about is how we move things out of the cell, and that's commonly called bulk transport. See, we're moving large things out of the cell, not individual molecules. So you'll see our bullet point labeled exocytosis. And on the right-hand side, I see a close-up view of a cell, I see its membrane, and I see its environment illustrated in blue. Inside that cell, I see a small spherical inclusion called a vesicle. And that vesicle is a membrane-bound structure that contains molecules. Follow the arrow, you see what happens is that membrane-bound vesicle merges with the cell membrane, fuses with the cell membrane, and at some point fuses with the outside environment, at which point it releases its contents to the environment. That is called exocytosis. That is a process how we're actually taking these molecules contained inside that vesicle and we're releasing them to the outside environment where technically those molecules have not passed through the membrane structure. My agent for getting those molecules out is the vesicle itself. So let's take a look at how we move things in. If you move things out, those cells are generally referred to as secretory cells because they secrete things. But on the other hand, if we bring things in, that's called endocytosis. And we're going to find there's actually 3 different forms of endocytosis that you are going to need to be aware of. The first is called phagocytosis. The prefix phago means to swallow. Cytosis means of the cell. So phagocytosis is cell swallowing. What we're going to do is we're going to bring in large solid particles into the cell through the exact same mechanism we just saw, except in reverse. Take a look at your illustration. You'll see a large pink cell with this plasma membrane illustrated, and you'll see a chunky piece of solid material on the outside of the cell. And I notice that there's this vesicle forming on the cell surface. That vesicle is an invagination, an in-pocketing of that membrane. And that particulate item, could be a food item, moves into that vesicle, at which point the vesicle moves into the cell and becomes isolated from the rest of the cell membrane. I have just brought a solid particle into the cell, technically without crossing the membrane structure. In some cases, what we can do is we can bring in large numbers of molecules. And by bringing in large numbers of molecules, what we're going to be doing is we'll be bringing in a large volume of extracellular fluid containing those molecules. Now, the illustration I want to refer to is the same one we started off with, and that's the one at top right. See, when we looked at the illustration at top right, we initially understood that that vesicle was essentially fusing with the cell surface and releasing its contents. Let's go ahead and play that in reverse. We play that in reverse, we get what's called pinocytosis. The prefix pino means to drink. Cytosis means of the cell, so pinocytosis is cell drinking. What we've got is we've got a significant number of molecules in the environment outside the cell. Those molecules plus a significant volume of the extracellular fluid flows into that invagination. And that invagination becomes isolated from the cell surface. And at that point, the molecules and the extracellular fluid are now inside the cell. So we're looking at the top right illustration, and we're reversing the arrows. We're starting with those molecules in the extracellular fluid. We're bringing them into the invagination, and that brings a volume of extracellular fluid into the cell. Hence the phrase cell drinking, the word pinocytosis. So we know about phagocytosis, cell swallowing. We know about pinocytosis, cell drinking. There's one more highly specialized version of this. It's called receptor-mediated endocytosis. And the question would be here, if I've got a large number of molecules outside of the cell, how can I grab hold of and import a certain select group of molecules without bringing them all in? That's why we're going to call this receptor-mediated endocytosis, because on our cell surface, there can be proteins that specifically bind certain molecules. If I take a look at our artist's illustration, you'll see what looks like a little letter Y located on the surface of the cell. And those little letters Y are proteins, and they specifically bind molecules in the extracellular fluid. In this case, I see those molecules are those purple triangles. If I go ahead and invaginate, do exactly what I've done the last two times, invaginate that membrane, and bring in those captured molecules on those receptors, I'm specifically targeting those molecules and bringing them into a vesicle. And so you'll see what amounts to being a invagination there coated with these receptors. Those invaginations are commonly called coated pits. And a coated pit is brought in with its bound molecules and becomes isolated as a coated vesicle, which is just one more way I can get something into the cell, but in this case, identify exactly what I want and use my receptors to do that. Be sure you know that bulk transport is moving large things. Be sure you know it's moving large things without crossing the actual structure of the membrane. That vesicles will be involved. Make sure you know exocytosis and 3 different types of endocytosis, phagocytosis, pinocytosis, and receptor-mediated endocytosis, and know the differences between each.